All right, we're going to very briefly talk about topics 2.9, 2.10, and 2.11. Now, topic 2.9 is mechanisms of transport, and this is really reiterating what we just talked about in topics 2.6 and 2.7. So, the enduring understanding is that cells have membranes that allow them to establish and maintain internal environments that are different from their external environments. So, you essentially need to know that we have a variety of processes that allow for the movement of ions and other molecules across membranes, which include passive and active transport, endocytosis, and exocytosis. So you can go back and revisit 2.6 and 2.7 to really look at the details on how we talked about passive transport, active transport, endocytosis, and exocytosis. So that's pretty much 2.9 is just mechanisms of transport. So looking at passive, active transports, endocytosis, and exocytosis. All right, topic 2.10, compartmentalization. So we actually talked about this topic within our 2.1 and 2.2. When we were going over our subcellular components, we really ended up talking about compartmentalization right there. So we're working off the same enduring understanding that our cells have membranes that allow them to establish and maintain internal environments that are different from their external environments. Now, our objectives on 2.10 is being able to describe the membrane-bound structures of the eukaryotic cell and being able to explain how internal membranes and membrane-bound organelles contribute to the compartmentalization of eukaryotic cells. Now, we've looked at all of this when we did 2.1 and 2.2 and we started going into each of our subcellular components. We ended up talking about all of this because it really just fell in right there when we were learning about our subcellular components. The essential knowledge that you need to know is that your membranes and membrane-bound organelles in eukaryotic cells, they end up compartmentalizing the intracellular metabolic processes and specific enzymatic reactions. And remember that this compartmentalization does come in eukaryotic cells. Okay, so they end up compartmentalizing the functions between the different organelles to make the cell work more efficiently. So it ends up compartmentalizing the metabolic processes that are happening inside the cell, as well as the enzymatic reactions that are happening within the cell. The other essential knowledge that you're going to know for this topic is that internal membranes facilitate cellular processes by minimizing competing interactions and by increasing surface area where reactions can occur. So these internal membranes that we have within our cell, they're really helping all of our cellular processes because we're not having the same job being done by different cellular components. Okay, so each subcellular component has its own job that it's contributing to the cell. So there is no competing interactions within our cellular components. The other thing is that we're increasing surface area where reactions can occur. So think about the mitochondria. We have increased the surface area within the mitochondria, and that allows more ATP to be made. Okay, so if this seems a little new or if you're unfamiliar, go back and watch the video when we did our study sheet on 2.1 through 2. All right, topic 2.11 is the origins of cell compartmentalization. So again, this is compartmentalization is what we've talked about already when we looked at our 2.1 and 2.2, but for this subtopic, we're coming in and we're looking at like evolution. Okay, so we're making that connection to evolution, where evolution is characterized by a change in the genetic makeup of a population over time and is supported by multiple lines of evidence. So, cell compartmentalization, ends up being one of those lines of evidence that support our theory that evolution is that change in the genetic makeup. So when we looked at the endosymbiotic theory and how our prokaryotic cells ended up being a major component of building our eukaryotic cells. So when we looked at the chloroplast and the mitochondria, and the evidence that we have that the chloroplast and the mitochondria at one point must have been prokaryotic cells, and when they became parts of larger structures or larger cells, we ended up developing our eukaryotic cells. So all of that comes into this topic where we're looking at the origins of cell compartmentalization. So your learning objectives for this subtopic is you should be able to describe similarities and or differences in compartmentalization between prokaryotic and eukaryotic cells. So right here, this is where we look at how prokaryotic and eukaryotic cells differ and how their compartmentalization is different or similar. So we know that, eukaryotic cells definitely we will see a lot more compartmentalization within our eukaryotic cells because remember that your prokaryotic cells are much, much smaller. Eukaryotic cells are larger than prokaryotic cells, and a part of them being larger means that they really need that compartmentalization in order to complete all their functions. Now, at one point, it was believed that prokaryotic cells did not have compartmentalization, but when we had the discovery of carboxysomes and metabolosomes, that was where they saw that there is some compartmentalization within prokaryotic cells as well. Okay, so that when they started looking into that, they noticed that there are bacterial microcompartments within our prokaryotic cells. And mostly these microcompartments or this compartmentalization is going to be within our protein or a lipid layer, but there is some compartmentalization in prokaryotic cells. So when you're describing your similarities or differences within them, you can't just say that we only have compartmentalization in eukaryotic, we don't have any in pro, because that would be inaccurate. You would have to go into talking about how it differs, where eukaryotic has much more compartmentalization and prokaryotic has very little, but there is some there. And then the similarity would be how compartmentalization is helping prokaryotic and eukaryotic cells be more efficient, that they're able to complete all of their functions using the compartmentalization. All right, now the essential knowledge that you need to know for this learning objective is that our membrane-bound organelles evolved from once free living prokaryotic cells via endosymbiosis. So this takes us back to our notes when we looked at the endosymbiotic theory or endosymbiosis theory, and we looked at our prokaryotic cells and we talked about how when our mitochondria and our chloroplast became a part of a larger cell, that's where eukaryotic cells appeared. So this is just that same concept. And you have to know that prokaryotes generally lack internal membrane-bound organelles, but they do have internal regions with specialized structures and functions. So think about your carboxysomes. In bacteria, we have these carboxysomes, which are bacterial microcompartments within the bacteria, and they are they're the predominant enzyme. They're working on carbon fixation. All right. The next learning objective that we have within 2.11 is describing the relationship between the functions of endosymbiotic organelles and their free-living ancestral counterparts. So when we looked at our notes, we talked about both the mitochondria as well as the chloroplast. So I want you guys to refer back to those notes and look at how we looked at the evidence within each of these subcellular components that, supported the theory that they were once prokaryotic cells. At one point, these must have been prokaryotic cells that eventually became parts of larger cells and became subcellular components of eukaryotic cells. All right, so remember that your mitochondria has its own DNA, and because it has its own DNA, that supported that theory that this has its own DNA that is separate from the DNA of the rest of the cell. And the same thing with chloroplast, the DNA that is found in chloroplast is actually very similar to bacterial DNA. And that again supports that theory that this must mean that chloroplasts were once prokaryotic and they became parts of a larger cell, becoming subcellular components of a eukaryotic cell. All right, so the essential knowledge you need to know is that your membrane-bound organelles evolved from previously free-living prokaryotic cells via endosymbiosis. All right, so that is pretty much everything for 2.11, 2.10, and 2.9. So I definitely suggest you go back if there are components on here that you are not familiar with or you didn't fully understand. I would like for you to go back to your notes when we talked about these topics earlier, embedded in our other topics, so that you can make sure that you fully grasp the essential knowledge and the learning objectives within these topics.